All right, so Dr. Borghammer is a leading expert in clinical imaging and neurodegenerative disease research with a focus on Parkinson's disease. He is a professor of nuclear medicine and neuroscience at the medical faculty of Aarhus University in Denmark. Dr. Borghammer explores how Parkinson's disease begins and progresses with a special focus on the early stages. His research investigates whether the disease may, in some cases, start outside of the brain, such as in the gut, before spreading through the nervous system. Using advanced brain imaging, laboratory research, and animal models, Dr. Borghammer studies on how Parkinson spreads and why it affects people differently. His team is also developing new scanning tools to help detect the disease earlier and better understand non-motor symptoms such as sleep disturbances and digestive issues. His work is funded by leading organizations including the Lundbeck Foundation, the Michael J. Fox Foundation, and the Danish Research Council and Danish Parkinson Association. And now I would like to welcome Dr. Borghammer.
Thank you very much, Kelly, for that nice introduction. So I'll just see if I can share my screen and the slides. Let's see. All right. I hope you can see my screen. Okay, so thank you very much for the kind invitation to give this presentation. Yeah, so first I'll be speaking for 30 minutes, and then there'll be a 5-minute break where something interesting will happen. And then after the break, I will talk a little more. And as you can see, the program is, first I'll give an introduction to what a theory we have hypothesized in my group called the brain first and body first hypothesis. Then I'll talk about various kinds of evidence, from clinical evidence, post-mortem studies of brains, imaging, and a little bit of animal perhaps. And then after the break, I'll just talk for a few minutes about what could the possible causes or triggers of these different types of Parkinson's disease be. But excuse me, I will start here. I'm sure many of you have heard of the Bragg hypothesis, postulated by the German neuropathologist Heiko Bragg 20 years ago. And it's a very important idea because he postulated that in Parkinson's disease, the pathology starts basically in a single place and then spreads from there. What he said is, in this red dot, he said, well, in fact, he said it starts in 2 places. It starts in the nose, in the olfactory nerve, the smelling nerve. But that's not important because it doesn't spread anywhere, he said. So the important point is that it seems to start in the gut, spread through this nerve, the vagus nerve, to the bottom of the brainstem, and then up and up from there. So that was kind of really a revolutionary idea. It kind of fits with at least some clinical evidence, because it's known that some patients with Parkinson's disease develop constipation years before diagnosis. So it makes sense that the pathology started years before. So that's why you get this early symptom. Some patients get REM sleep behavior disorder before diagnosis. And this comes from the pons here, which is below the dopamine system. That's why it comes before. And then when the disease hits the dopamine system, that's when you get the diagnosis. So that was the idea. But we have known for many years, basically, that it is an incomplete hypothesis for many reasons. And I will only mention 2 of them. First of all, we know that only approximately 1/3 of people with Parkinson's develop constipation and REM sleep behavior disorder before diagnosis. So only 1/3 have these symptoms. And if Bragg was right, we would expect that all of them would have it before diagnosis. But more importantly, and this is a very busy table, but you should just look at these 25 zeros here, because this table shows 25 cases, brains from patients with Parkinson's disease. And these cases did not have any pathology in this place where Bragg said that it always started. So clearly these patients did not fit the Bragg hypothesis. So based on that sort of thing, postulated the body and brain hypothesis a few years ago. So body first disease is very similar to what Bragg was saying. The pathology in most people probably starts in the gut, in this subtype, goes through the vagus nerve here, but it also goes through another system, the sympathetic system, it's called, and that's the route by which it reaches the heart very early in this type. And then it basically goes up and up through the brain, as Bragg said. But this is only one type, the body first. The brain first type is a type we believe that in most people actually starts in the nose, hits the olfactory nerve, spreads very quickly to a place called the amygdala, which is just adjacent to the dopamine system in the nigra. But then in this type, the pathology then spreads in the other direction and eventually also reaches the organs of the body. So that means that, in late stage disease, 20 years after diagnosis, perhaps, patients are more and more similar, and they start to get the same symptoms, but very early on, they are very different. Okay. I think I'll skip that for the moment. So here, when we look at patients with Parkinson's disease, we believe that only perhaps 1/3 of them have the body first type, where it perhaps started in the gut, whereas perhaps 2/3 have the brain first type. But if we look at a related disease called dementia with Lewy bodies, we believe it's the opposite. About 2/3 have the body first and 1/3 the brain first. So why do we believe that? The point is, here in green, this is the sleep center, right? And in the body first type, they get the sleep problems before diagnosis, right? Because the pathology comes from downstairs. And we know from many, many studies that only about 1/3 of Parkinson patients get the sleep disorder before diagnosis. So this suggests that only 1/3 have the body first type. When we look at dementia with Lewy bodies, it is at least 2/3, perhaps up to 80%, that develop the sleep disorder, suggesting that many more of dementia with Lewy bodies are the body first type. Also, when we look at other symptoms, for instance, constipation here, and this is actually the time before diagnosis, and you can see dementia with Lewy bodies in red here compared to Parkinson's. Dementia with Lewy bodies have more constipation up to 14 years before diagnosis. They also have more hypotension, problems with their blood pressure up to 8 years before diagnosis, and more urinary problems up to 6 years before diagnosis. So dementia with Lewy bodies they have a lot more bodily problems, if you will, 5, 10, 15 years before diagnosis, suggesting that they are more of the body first type. Okay. So again, looking a little bit at the clinical evidence, so a graph like this, I'm sure you've seen it many times. If you just want to show one figure and describe what is Parkinson's, this is the figure that you see. And it shows you that in Parkinson's disease, you get constipation and REM sleep behavior disorder before diagnosis. Excuse me, I think I'll just see if I can get a little laser pointer here. There you go. So REM sleep behavior 10 years before diagnosis. But the point is, this way of showing the disease is only appropriate, you could say, for 1/3 of Parkinson's, because as I said, only 1/3 develop constipation and REM sleep behavior before diagnosis. And we believe this is the body first type, because these are kind of symptoms that come from below the dopamine system. So what you need to do, you also need to show this graph actually, showing basically the disease course of many patients with brain first. And in these patients, they actually have quite few and quite mild symptoms before the Parkinson diagnosis here, but then they can develop the same symptoms, but in these patients, it comes after diagnosis. Okay, and if we put these two related diseases, Parkinson's and dementia with Lewy bodies, into one pot, and look at the clinical evidence, we would say about 50% are body first, follows this trajectory, and about 50% are brain first following this clinical trajectory. These are kind of ballpark numbers, but just an estimate. Okay, so that was a little bit about the idea, the clinical evidence. So let's look at some post-mortem evidence. So that's data from where brains and other organs from dead people with Parkinson's have been examined. All right. So you see the figure on the right here, and all the red dots, they symbolize pathology. And the point here is that once a patient gets the diagnosis of Parkinson's or dementia with Lewy bodies, the pathology is quite widespread. It's almost reached everywhere where it will eventually be at the time of diagnosis. So that means if we want to guess, so where did it start? It's a little difficult to guess because the pathology is also already so widespread. So therefore, it's incredibly important to study what we call incidental Lewy body disease. And this is just, you know, brains from brain banks where there's only a little bit of Parkinson pathology. It hasn't spread very far, because if it hasn't spread very far, it's easier to guess where it came from. So I did a study we published a few years ago with some international pathologists, and we examined 528 brains from people that had Parkinson pathology. But among these, we isolated the approximately 1/3, 155 cases. Those were the cases with the earliest pathology. They only had a little bit of pathology. And when we did that, we see that basically two patterns emerge. Either the pathology is only at the bottom of the brainstem, but also in the peripheral organs. In these patients, it's not in the top of the brain. So we think this is body first, early body first. In the other type, it's in the olfactory nerve and some other places close to that. But here, it's not below that. It's not in the organs. It's not in the bottom of the brainstem. And I'm going to go through this very rapidly. But all these colors show you different cases with the disease. The only important point is that all these people up here, they had disease in only red and yellow regions. So that is here, but they had absolutely nothing in blue. Here you see other cases, they only have the pathology in blue and yellow regions. So those are these cases. They have pathology here, blue and yellow, but then they did not have anything in the red regions. And then only a couple of cases, 7% did not fit. Another data set, a Finnish data set, is the same thing, red and yellow, blue and yellow. Only 5% didn't fit. And yet another large Finnish data set, red and yellow, blue and yellow, and only 8% didn't fit. So in 3 different large data sets, we see the same thing. And when you boil it down, what we found is that approximately 46% of all early cases look like this, compatible with body first. 47% look like this, compatible with a disease that came in through the olfactory nerve in the nose. And then there are rarer cases, 7%, that do not fall into these 2 categories. Okay, so of course we believe that this is an argument to say that the earliest pathology in Parkinson's disease either comes in from the nerves of the body, and that's why it shows up down here in almost half of the patients. In the other half, it seems to have come in via the nose and then to the adjacent regions. Okay, this is another similar study we did in collaboration with 2 brilliant Japanese professors in pathology, and these are my 2 students, Katrina and Anushri, who did the study. So here we took 173 cases with Lewy body disease and put them into a computer program, a machine learning algorithm. And this algorithm kind of tries to guess where did the disease start and how does it spread. That's what this program was built for. And this program the output was, okay, so about 50%, 51% is compatible with brain first. The program suggests that it starts in the nose, goes to the amygdala quite close, and then to the dopamine system and a few other regions. So that's completely compatible with the brain first, I said. And then it said, okay, so the other half is compatible with body first. But here, a surprising thing was that it said it's actually two things. It's two different body first types. One of them goes mainly through what we call the vagus nerve from the gut via the vagus nerve directly to the brainstem. And so it's quickly in the brain and then starts going up. And then it goes to the heart later. That's what we call the parasympathetic type. The other type, it seems to stay much more in the body for a longer time. So it goes not to the brain directly, but it goes closer to the spinal cord and early to the heart, and stays there for a little while, perhaps years, in fact, before it eventually goes to the brain. So that's an interesting new finding that we are pursuing further, and I'll come back to it in a little while. And it actually makes sense that there are these two types, because when we look at our early body first patients, some of them, they seem to have some, problems with the blood pressure and other things, suggesting that there's something wrong with the sympathetic system to the heart and the vessels. But other of these sleep patients, these early body first, they don't have these symptoms at all. So there seems to be some variance even in this body first group. All right. So now that was the postmortem. Now I'll talk a little bit about some imaging evidence. And I'm only basically going to talk about two modalities. Heart imaging. So we have a scan that can image the heart. This is the heart. And what we are imaging here is the sympathetic nerves to the heart. That looks like this in a normal person. And then we also image the dopamine system with PET scans that I'm sure everybody is familiar with. And basically what we were looking for, the hypothesis was this, and it's also what we found, but I will explain it here. Very early brain first patients, they are only sick in the brain, so they should have a pathological dopamine scan here. But if they are early, the body is still normal, so the heart is still normal, as you can see here. That's brain first. In very early body first, we expect to see the exact opposite thing. The body organs are sick, if you will, so the heart scan is abnormal. You can't see the heart anymore, but the brain is still normal. So here you can still see a completely normal dopamine system. So it's completely the opposite pattern. And that's what you see here, lots of data. The dopamine is down here. This is the brain axis. The heart axis is here. So when you're in this top right corner, you're normal on both scans. And these are our healthy control persons we imaged. Blue is body first. And you can see these patients started by going down. So they first lose the heart fibers, but the brain is still normal because they're still to the right here. They have completely normal scan. But then they start moving left, and eventually they also start to lose the dopamine innervation. And of course, that's when they get the Parkinson diagnosis. But all the red patients are brain first, and they have the opposite trajectory. They start by moving left, losing the brain dopamine system first, but they still have a normal heart. But then eventually they'll start to lose the heart fiber innervation. And the red patients are the sort of patients that have no sleep problems, no constipation. They didn't have much symptoms except perhaps loss of sense of smell. But so this is what they look like on this imaging. All right. So these two trajectories that almost don't overlap with each other, they really powerfully suggest that this theory is correct. Either you see damage to the brain first, or you see damage to the body and the heart first. And therefore it fits with the brain first, body first idea. And this is a picture of some group members that mainly collected all this data, because studies like this are tremendously complicated to do. And also, of course, I'm very grateful to all the patients who contributed. Without their help, we would never be able to do this kind of study. I'll skip this in the interest of time as well. Okay, now I decided to take this because I know from my American Canadian colleagues that these heart scans are not so familiar to people in North America because it's not used so much as it is in Europe and Japan. So again, all of these dots here are Parkinson patients. And up here at this line here, this is the level of normal people, healthy controls. So you can see some Parkinson patients have normal levels, and they look like this. You can easily see the heart. When you have lost 50% of the fibers, you can still see the heart here, but now the heart is the same level as the lungs. You can see the big patches here, whereas up here it was way more intense than the lungs. And then when you have lost everything, 100%, it's completely invisible. Okay, but the point here is that this is very slow. It does not happen overnight. So it takes about 11 years to going from normal to having lost 75%. How do we know this? Because, yeah, this is irrelevant for you, I guess. We're going to publish this later, but we, in collaboration with Korean scientists, we have studied 200 of these scans where the same patient had several scans over years. So therefore we can estimate how fast does this go. And we found that it takes around 11 years to lose 75%. And so coming back to this was the picture from the last slide. Remember, these are healthy controls with normal hearts. And at this line here, these are the sleep patients. And at this line, they have lost 75% of the heart fibers. And we now know that this takes 11 years. So it's very, very slow. This is a powerful argument for calling it body first, because this heart problem has been going on for 11 years, but the patient still has a normal brain. Once they are at this level, we know that it'll take another 8 years before they actually converge and get the diagnosis of Parkinson's. So this is another way to kind of demonstrate how relatively slow a disease, if you will, Parkinson's disease is. So all of this that we show in this graph here, all of that happened before a body-first patient even got the diagnosis of Parkinson's disease. Yes, I think I will skip this as well in the interest of time. Okay, moving on to this one. So this model, this theory we have proposed, we believe that it can also explain, at least to a certain degree, why is Parkinson's disease sometimes an asymmetric disease where one side of the body has more symptoms than the other. And when we look at a PET scan of the dopamine system, we can see this asymmetry. In this patient here, we can see that there's more dopamine lost on this side than on this side. It's more asymmetric. But we also know that some patients with Parkinson's, they actually have a quite symmetrical scan. And according to our theory, and I'm not going to explain to you why, just briefly say that the theory postulates that it's mainly the brain-first cases that are asymmetric, whereas the body-first patients, where it came from the gut, is more symmetric. And I just want to remind you that at the beginning of my talk, I said that dementia with Lewy bodies, a similar disease, they are more body-first. So dementia with Lewy bodies should be more symmetric, whereas Parkinson patients on average should be more asymmetric. And again, that's what we found. These are all dopamine scans. And these colors, it's what we call an asymmetry index. Healthy controls have normal scans, very symmetrical. So they have a blue value here. They have a symmetric scan. These are body-first Parkinson's. Most of them have symmetric scans because they have lost both sides of the dopamine system. Whereas most of the brain-first patients here, they have much more asymmetric scans, tending to look more like this with more asymmetry. This was published 4 years ago. This is new data, not published, but it shows the same thing. Most of the brain-first patients have more asymmetric scans up here. Most of the body-first patients have more symmetric scans. And finally, dementia with Lewy bodies are more of a body-first type. In this study that we published, 80% of them have symmetric loss, when we compare to a lot of early Parkinson's, 2/3 have an asymmetric loss. So everything I show here fit with the model, the brain first, body first model, and also seems to be able to explain which groups are more symmetric and which are more asymmetric. Okay, I think this is my last slide, the next slide before the break. So I just want to say we're also doing various animal studies in order to try to study the disease closer and to develop some models where we can test drugs that can hopefully benefit the patients. So this is just one slide. We're trying to simulate brain and body first in rats. So here in body first, we start the disease in the gut, and we see then it spreads through the sympathetic system, and it reaches the heart. You can see the pathology here. It goes through the vagus nerve to the bottom of the brainstem, and it also reaches the top and the dopamine system. So this is the body first animal. The brain first, we start the disease close to the smelling nerve. Then it spreads quite quickly to the dopamine system, and then it starts to spread in the other direction, reaching the gut. So the spreading pattern is the same as we see in humans. Now, what about, for instance, if we look at the heart? Because remember, brain first patients have a normal heart early, whereas early body first patients have a pathological heart. And we see exactly the same thing in these animals. These are two control groups. The red dots are brain first animals. They have completely normal heart. The blue dots are body first animals. They basically have an invisible heart, like in the human patients. That's what we show here. And again, about the asymmetry, remember, brain first have more asymmetric scans. This is the brain first scan of the rat. And in a human patient with brain first, it looks like this, asymmetric. This is the dopamine scan of a body first with symmetric loss. And in a human body first, we see the symmetric loss. So these animals seem to quite closely look in the same way as the brain first and body first patients. All right, I think, I should mention, of course, that my good colleague Natalie Vandenberger is doing all of these animal studies. So that is the 5-minute break. And I suppose I'm going to de-share my screen here.
Thank you, Dr. Borghammer. And now I would like to welcome Rosemary from Improv for our 5-minute movement break.
Yay! Hello. Kelly, really quickly before you go, are the attendees of the Zoom able to use the chat or no? Can anyone see this?
Yes, they are.
Amazing. Okay, hello, folks who are attending this webinar. My name is Rosemary. I work with Improv for Work and Wellness, and we have an Improv for Parkinson's program. So if you don't know what improv is essentially spontaneous theater, making things up together. It's a lot of fun. We use it for Parkinson's primarily as cognitive exercise, so exercising the brain, and positive social connection between the Parkinson's community. So I have a couple of games for us to try on our 5-minute break, and I'm modifying them because I can't see any of you in front of me, but we're going to give this a try. So one of the first games that I love to teach with Improv for Parkinson's and a lot of our Improv for Wellness programs is a game called That Makes Me Think Of. So this is just a word association game. I was inspired to do kind of one more in the brain and one more in the body. So this is a little bit of a brain game. The word association game is just, we would usually go around in a circle maybe, where one person would start off with any word at all. And the next person would take that word and say, that makes me think of, and another word that it makes them think of. For example, it might go something like poster. Poster, that makes me think of painting. Painting, that makes me think of paintbrush. Paintbrush, that makes me think of artist, and so on and so forth. So I'm going to modify this for us in the Zoom webinar format. I'm going to start off with a word, and I'm going to first see if people will respond in the chat. So I'm going to put out a word, and I just want you to think of the first thing that comes to your mind. And in this one, this is not like therapy. There are no wrong answers. You can say whatever. So the first word I'm going to put out there is lamp. Lamp. Putting this into the chat. Light. Amazing. And if other folks want to throw things in, and what I'm going to do is I'm just going to jump back and forth between myself and the chat. So great. Oh, okay, I'm going to take street. Street makes me think of cars. Now I'll go back to the chat. What does cars make you think of? Engine, fast, Uber, yes, yes. Okay, I'm going to take engine. Engine, oh, engine makes me think of steam cars, you know, those kind of like old school cars. What does a steam car make you think of? Train, driver, yes. Energy, yes. Amazing. Okay, energy. Energy makes me think of running. Running. What does running make you think of? Throwing back to the chat. Track meet. Yes. Fast, tired, healthy. Yes. Yes. Awesome. Okay. One more. Oh, stamina. Okay. I'm going to do one more for this one. Stamina makes me think of, oh, train marathon. Stamina makes me think of triathlon. I want to do a triathlon. Stamina makes me think of triathlon. What does triathlon make you think of? Back to the chat. Swimming, too long, yes. Crazy, yes. Olympics, exhausted, cycle, run, swim, very sporty. I love these answers, amazing. Okay, so that makes me think of, it's a word association game. We play this often as a starter because with Improv for Wellness, it's not about being funny, it's just about connecting and working our brains. And even though you all are chatting with me through the Zoom chat, I still feel like I got to know you a little bit just by where your brain goes for these answers. You know, like triathlon, and somebody commented crazy. That was the first thing it made them think of. I love that. Amazing. Okay, I'm gonna go quickly to our next game. This is a similar type of one. This is a body first game, stealing the body first, brain first. We often in improv forget that we can use our body to inspire our brains. So we like to incorporate that a lot in Improv for Parkinson's. This is a game called Try That On for Size. And we often play this as a competition at an improv show, but we're going to modify it again for the Zoom format. So I'm going to start off with an action. I'm just going to start off like this. And if you can see this, you can copy me. This is the action. We'll start like this. And then all we're going to do is justify the action of what it can be. You don't have to type it in the chat yet. I might ask you for answers afterwards. But what I'm going to do is I'm going to go back and forth, and I'm going to say what this might be. And then I'll leave a pause, and you in your Zoom room, wherever you're watching this, you can also justify something different than what I said. So I'll start off and I'll say, I'm waving my hands. Try that on for size. And then in the Zoom room, you would say, oh, jazz hands. Yes, amazing. If you want to comment what they are, absolutely. And if you're not sure, sometimes just doing the action is what helps. Windshield wipers, I love it. I'm casting a magic spell. Try that on for size. I'm throwing it back there, shaking. Yes, amazing. Okay, I'm doing another one. What are all of the things that this action could be? And if you can't think of it, try it first and then justify. We often go back. Tumbling, yes. I'm making pizza dough. Try that on for size. Oh, shadow boxing. Roly, I love. Reeling in a fish, yes. Okay, something that people haven't said yet. I'm tying a bracelet around my arm a bunch of times. rolling yarn. Yes, yes, amazing. Okay, and we'll do one more. Okay, what is the thing I want to do? Oh, something different. Amazing. Okay, this is our very last one. I'm saying hi to a little bird. Try that on for size. And a worm. Yes, I love it. Yes, try this where you are. Trying to talk. Yes, raising my hand on Zoom. Duck. Try that on for size. I love it. I'm doing finger exercises. Try that on for size. Amazing. Okay, that was a little bit of a brain workout, a little bit of a body workout that's also working our brains. Thank you so much, everybody. Yay!
Thank you very much, Rosemary. That was very great. I'm sure everyone enjoyed that. And now I will pass the floor back to Dr. Borghammer.
Okay, thank you very much. That's absolutely the best way I've ever been interrupted in one of my talks. Great, I loved it as well. Okay, so yeah, so as I said, I have just I think 6 or 7 slides here, and this just to talk a little bit about. Okay, so now we established that it seems plausible that we have kind of brain first disease coming through the nose, perhaps body first coming through the gut or other organs. So we don't know enough about what the triggers of these things, so these two types might be, but it's something that's being explored a lot, internationally, in many different groups. Last year, I wrote a paper along with Ray Dorsey that some of you may know. He authored the Ending Parkinson's Disease book and then published another book recently, The Parkinson's Plan, along with Professor Michael Oken. So we wrote a paper with Ray, and also Brianna G. Miranda was on it, and one of my colleagues, Jacob Horsager. And the idea, well, basically of Ray's books, I should say, is, Ray is really an advocate for the idea that Parkinson's may be an environmental disease. So we basically try to write a paper coming up with ideas, a research framework, if you will, to study more closely how that might be. So all kinds of industrial toxicants, pesticides, and other things. Either you could inhale them, that brings them directly in contact with the olfactory nerve, and then it would basically potentially trigger a brain first disease in your olfactory nerve, which is just very, very close to that place. Everything you inhale basically ends up in your gut because we continuously swallow the secretions in our nose. So the same toxin would actually also end in the gut. On the other hand, if you drink or eat toxins of some sort, or pesticides in drinking water or whatever, it could go directly to the gut, start a disease that then spreads through the vagus and so on. So we speculate about these things. Just one more point here. So for instance, pesticides, if you're a farmer or a gardener and been exposed to a lot of pesticides in your work, a lot of that might come onto your skin, which potentially could actually trigger a skin first Parkinson's, because that would actually tend to spread to the same sympathetic system that connects to the heart. So that would not be a gut first, but a skin first disease. But that might actually not be too different from a gut first. It would end up being a body first type. Yeah. Now that was pesticides and toxicants, but basically exactly the same thing could be going on, about virus, bacteria, diseases with inflammation, and so on. Because that they would tend to use the same entrances. You can have respiratory infections in your nose or lungs, or you can have gastric, and gut infections and so on. Okay, so what are the, so I think perhaps if we focus mainly on body first for the remaining 5 minutes. So what are could the causes of body first, gut first Parkinson's be? Well, we already know in registry studies that inflammatory bowel disease like Crohn's disease, colitis, that increases your risk of Parkinson's. We also know that repeated gut infections increases the risk. But in the vast majority of these studies, they cannot separate brain and body. They don't have that information, so we don't know. However, there are some studies suggest that pesticides in itself, at least pesticides, I think we know, that increases the risk certainly for body first disease. Yeah. Now this is a very busy slide. It's about microbiome, you know, the bacteria and so on in your gut. Tremendously complicated. So this is a very simplified version of such a data set. The point is, here are some healthy controls, here are some Parkinson patients, and down here, so each column is a patient. So if we focus on this red box here, these colors depict some bacteria types. And here we have the patients, and remember we have brain and body first. And actually the point is the black box here signifies the body first, because they had the sleep disorder before diagnosis. And you can see they have different bacteria here. They have these colors here, whereas the brain first, they look like the healthy people. So that's quite interesting. It seems like there are some bacteria here that are specific to body first. And that's been seen in other studies. This is a study only where they studied the sleep patients, so that's before diagnosis. So that's Parkinson's disease, body first before diagnosis, called RBD. And again, you can see there is some overlap, but certainly these patients have a different, they have different microbiome, different bacteria compared to healthy people. Okay. We also know, for at least from a few studies, that it seems like some Parkinson patients have a leaky gut. Again, a busy slide, but on the left here, when your gut is healthy, these are the cells, the wall, the barrier in your gut, and they are glued together with something called tight junctions. So nothing can get through, basically. When you have a leaky gut, these cells are not, they slip apart. These tight junctions are not so tight anymore. And therefore bacteria, virus, and so on can actually access your gut. And so this brown color that you see, these are images of a gut seen in a microscope. And the brown color is actually a color that comes from bacteria. And it shouldn't be, the bacteria shouldn't be inside your gut wall. They should be outside in the gut contents. So in some Parkinson's, you see this brown, but you don't see it in healthy control, suggesting that some bacteria has managed to gain entry to the gut wall, if you will. Right. I guess this has to be my final point in the interest of time. Another interesting fact about some bacteria, they can produce a protein called curly. This is biofilm, because seen in a microscope, some bacteria, they envelop themselves in slime that we call biofilm, and they use this curly for this purpose. But it was discovered that when you take curly and you put it in a test tube into where you have some normal alpha-synuclein, that's the protein that gives you the Parkinson pathology, then basically this protein starts to clump together in bigger and bigger clumps and forming something that looks like Parkinson pathology. And remember, it was triggered by a protein that came from a bacteria, right? Well, I'll skip that. Okay, so that means theoretically, it might be some of our gut bacteria, they produce the curly, okay, but it cannot enter the body under normal circumstances. But if you have a leaky gut that some Parkinson patients have, then this protein can enter. It gets into contact with the normal alpha-synuclein protein, which we know from the test tube can make that aggregate into clumps. The clumps will be bigger and bigger, and maybe that is one way you can get a Parkinson's disease started in the gut. But most people have this protein in the gut. Not everybody gets Parkinson's. So you definitely need to have more factors, perhaps some vulnerable genes. Also, the older you get, the less your body is able to defend itself against these mechanisms. And if you get some repeated gut infections on top of it, that might also increase your risk of getting this bad process started. Right. So this is my summary slide. Just to say here in pictures, I told you about the concept of body first and brain first. When we look at the clinical symptoms of patients, it also seems like we have two overall categories, if you will. One type of patient has some bodily symptoms, autonomic problems, constipation, sleep problems before diagnosis, suggesting they are body first. Other patients do not really have any, suggesting they are brain first. I showed you our imaging data that we can separate these two types into two groups that almost don't overlap if we find the patients early enough. When we look at post-mortem data from brain banks, we see that approximately 50% are compatible with brain and body first. And finally, here after the break, I talked about just a few quick ideas about what we're doing research in right now about trying to study what might some of the triggers be. We need to learn that, also to be able to prevent future Parkinson's, at least some cases, by preventing some of these toxins that might be responsible for causing it. So with that, I would like to thank you especially for your participation, and I most especially would like to thank the patients in Denmark that have contributed to our study and to the increase of knowledge that we have been able to produce. So thank you very much for your participation.